rest of the part of the GIT, of development of GIT. So how far we have reached? So you know the derivatives of foregut. So most of the derivatives of foregut we have discussed in the previous session. And one thing more you should remember, and that is the major digestive glands, that is the liver. Then what else? Gallbladder, pancreas, extrahepatic biliary apparatus, all those things, all those major digestive glands, they are developing from actually foregut. So in this session, we'll look at the other derivatives of foregut. So first is the development of liver. So where does this liver develop from? I told you it is actually a derivative from foregut, and that is in the distal part of the foregut. So what you see here, this is the foregut region and this is the midgut. So you see in the distal part of the foregut, you find a small diverticulum is coming. Look at that. You find a small diverticulum, and that is what is termed as the hepatic diverticulum. Now what happens is this hepatic diverticulum will be actually passing through the mesentery and then proceeding on towards the septum transversum. That is how you get the proceeding of the hepatic diverticulum or endodermal hepatic bud. So first of all, it projects or it protrudes or passes through the ventral mesentery to the septum transversum. Now what happens is this hepatic bud now divides into a larger part termed as the pars hepatica and a small part termed as the pars cystica. Okay, so just look at those two names. One is pars hepatica, the other one is pars cystica. So now you can say the derivatives that is coming from pars hepatica and pars cystica. This pars hepatica will further divide into two buds, that is further or later forming the two lobes of the liver, or they branch further to form the lobes of the liver, and further they divide to form different cords of cells there, that is the cords of hepatocytes you find there. Then what else? In between that you find the bile canaliculi, biliary canaliculi, all these things, the hepatocytes, the two lobes that is formed inside the bile canaliculi, all these things are formed from the pars hepatica. Fine. Now what happens to the pars cystica? This pars cystica will form, this part will form the gallbladder. Cystica, you know, gallbladder, and the proximal part will form the cystic duct. Fine. And then what happens is this proximal part of this hepatic diverticulum will be further converted to common bile duct, and that common bile duct will be further coming and opening into the duodenum, in the second part of the duodenum, that is on the dorsomedial wall. Fine. So now look at it. You are seeing the hepatic diverticulum. You are seeing the hepatic diverticulum dividing into pars hepatica, pars cystica forming the gallbladder and cystic duct, pars hepatica further dividing into two lobes. From there you find the whole of hepatocytes, the lobes, the hepatocytes, the bile canaliculi, all these are developing from it. But don't think the whole of the liver is being developed from pars hepatica. No, it's not like that. The transversum has also got some contribution. What happens is, as this pars hepatica is proceeding through the mesentery to reach the septum transversum, what happens is, within the septum transversum, you find small vitelline veins and umbilical veins. They undergo breakage and then undergoes realignment so as to form the sinusoids of liver. Fine. So sinusoids of the liver are actually formed from this broken umbilical vein and the vitelline vein that is present where? In the septum transversum. Now what happens is, this fibrous capsule, liver has got a fibrous capsule, you know, termed as the Glisson's capsule. Then you have got Kupffer cells, hematopoietic cells, all those things are derived from the septum transversum. So these three derivatives should be there in your mind when you are thinking of the development of liver. Fine. So read that. The liver is actually an endodermal hepatic bud which arises from the ventral aspect of the distal part of the foregut. I've told you it is in the distal part of foregut. Then it is passing through the ventral mesogastrium to the septum transversum. It divides into pars hepatica and pars cystica. Pars hepatica later forms the right and left lobes of liver. Cords of hepatocytes are formed from there. Then what else? Not only the cords of hepatocytes but also the biliary canaliculae are formed. All these are formed from pars hepatica. Now what happens as these pars hepatica is passing and reaching the septum transversum, the vitelline vein and the umbilical vein undergoes breakage, which later forms the sinusoids of liver. So now you know the two derivatives. One is from pars hepatica, second derivative is from the broken vitelline vein and umbilical vein that is present in the septum transversum. Third derivative also you should remember. What is it? That is the mesenchyme of septum transversum. Septum transversum also has got a contribution. So it contributes in the formation of the fibrous capsule. Then what else? The Kupffer cells. Then the peritoneal covering, the hemopoietic cells, the blood cells. All these things will be derived from septum transversum. Fine. So this much things about the development of the liver you should remember. So in some applied aspect, one is Riedl's lobe. So what you see here is the Riedl's lobe, and commonly it is a very commonly seen. You can see as an extension coming from the right lobe, and in certain cases it could be mistaken for a tumor or something like that. That is what is termed as Riedl's lobe. Second one is polycystic disease of liver. Polycystic disease of liver. So what is that embryological cause that causes polycystic diseases? It is because of the failure of union between the intrahepatic and extrahepatic bile ductules. Once this connection is not established, that would lead to polycystic disease of liver. That is very commonly associated with polycystic disease of kidney. That figure, once you are seeing here, is the normal one. You are seeing a large number of cystic lesions in the liver. That is mainly because of this failure of connection between the intrahepatic and extrahepatic ductules. Then you have to remember intrahepatic biliary atresia. That's a very serious anomaly. Very commonly it goes for the death of the baby, or otherwise the baby have to go for a liver transplant. It is one of the very serious anomaly, intrahepatic biliary atresia. Next is Caroli's disease. Caroli's disease means it is a congenital dilatation of intrahepatic biliary tree. So this much about the development of the liver you can remember. So where is it actually developing? It is a hepatic bud developing from the distal part of the foregut, passing through the ventral mesogastrium through the septum transversum. So the liver is then it divides into pars hepatica, pars cystica. It's pars hepatica which further divides into two, which forms the two lobes of the liver. So pars hepatica forms the lobes of the liver. Inside that it undergoes small cords of cells. These cords of cells will be aligned to form the cords of hepatocytes. The bile canaliculae also is formed there. So that is the first source. Second source you have to remember what is it? It is from the septum transversum, which contribute towards the covering Glisson's capsule, fibrous stroma, hemopoietic cells, Kupffer cells. All these are derived from the septum transversum. Plus you have to think of that broken down vitelline vein and umbilical vein that is present within the septum transversum, which forms the the sinusoids of liver, plus some applied aspects of liver you should remember. So with that we finished the development of liver. Now the development of gallbladder. I think I've already discussed, just read that. It is actually formed from pars cystica, and what happens is it is the pars cystica which forms the gallbladder plus the cystic duct. Whole thing forms the gallbladder plus the cystic duct, and the part proximal to pars cystica that forms the common bile duct. So what happens to the common bile duct? It initially comes and opens into the ventral part of the duodenum, and then the duodenum is undergoing rotation, And as a result of it, this opening now moves on towards and reaches the dorsomedial aspect of duodenum. Fine. So now some anomalies of gallbladder. Agenesis. Sometimes the whole gallbladder could be absent. And next is sessile gallbladder. Sessile gallbladder, where cystic duct, this is a cystic duct there, this cystic duct could be absent. Cap. So look at that, this is a pharyngeal cap where in the fundus, this is a fundus of gallbladder, is twisted so as to form a cap-like projection there. That is what is termed as pharyngeal cap. Then Hartmann's pouch. This is the neck of the gallbladder. In the neck of the gallbladder, you find a small extension or small pouch termed as the Hartmann's pouch, and it is one of the very common site you get the gallstones could get dislodged or could be present there in the Hartmann's pouch. And in certain cases, you find the gallbladder could be septate, double gallbladder could be there. Sometimes the gallbladder could be intrahepatic. Sometimes the whole of the gallbladder will be covered with or will be whole surrounded by a mesentery. Then you call it as a floating gallbladder. A large anomalies in the ducts could be there in gallbladder. Large number of anomalies there can be mentioned for gallbladder. Okay. So that is about the gallbladder. So just a recap of the development of gallbladder. Develops from pars cystica. So pars cystica forms the gallbladder as well as the cystic duct. Fine. So this is the pars hepatica. This pars hepatica now have formed the liver. So this is the proximal part. This proximal part will form the common bile duct. Fine. So I think with that we finish the development of gallbladder and proceeding on towards the development of pancreas. So this is the foregut. This is the foregut. This is the midgut. So actually it is at the junction of the foregut and midgut you find the pancreas is developing. So first, just above, this is the dorsal pancreatic bud and this is the ventral pancreatic bud. So just above, when you compare, above you find the dorsal pancreatic bud developing, and this dorsal pancreatic bud will be actually present in the dorsal mesentery. Fine. Ventral pancreatic bud, when you compare it with the dorsal, will be a bit below, very close to your pars hepatica. Fine. This is your pars hepatica, this is your pars cystica. So very close to that you find the ventral pancreatic bud, which is actually proceeding through the ventral mesentery. Now what happens is this duodenum undergoes rotation. As a result of the rotation of the duodenum, what happens is this dorsal pancreatic bud now comes to lie on the left side and the ventral pancreatic bud now comes to lie on the right side. Fine. Now what happens is, these are, one is on the left side, there's a dorsal pancreatic bud is on the left side, ventral very close to pars hepatica is now on the right side. Now what happens is, with the differential growth of the wall of duodenum, what happens is this ventral pancreatic bud will again rotate or pass down and will reach just below the dorsal pancreatic bud. So look at that. Once there is a differential growth on the wall of the duodenum, what happens? This ventral pancreatic bud will come and lie just below the dorsal pancreatic bud. Then what happens is this dorsal and ventral pancreatic bud will undergo fusion to form your pancreas. So what happens? It is this dorsal pancreatic bud which forms the upper part of the head. Look at that. Upper part of the head, the neck, the body and the tail, all those regions. Upper part of the head, the neck, the body and the tail will be derived from your dorsal pancreatic bud. This ventral pancreatic bud will form the lower part of the head plus your uncinate process of pancreas. So that is how you find the different parts of the pancreas are developing. So look at that. This is after the rotation of the duodenum. This is the dorsal pancreatic bud. This is the ventral pancreatic bud. You find the rotation is occurring. and after this ventral will come to lie just below. So what happens? This dorsal will form the upper part of the head, the neck, the body and the tail. What happens to the ventral pancreatic bud? It forms the lower part of the head region plus uncinate process. Fine. Now what happens? Now you know the development of pancreas. You should also be familiar with how the ducts of pancreas are developing. So look at that. This is the dorsal pancreatic bud. So this dorsal pancreatic bud is coming and opening into the minor duodenal papilla. Fine. This is your ventral pancreatic bud. It is to the ventral pancreatic bud. I told you it is very close to pars hepatica. So what happens? You find the common bile duct also coming and opening there along with your ventral pancreatic duct into the major duodenal papilla. Okay. So now what happens? This ventral has undergone rotation here and reached just below the dorsal. And now what happens is you find an anastomosis occurring, an anastomotic channel occurring between the dorsal and the ventral. So now what happens? So this is your dorsal pancreatic bud. You find an anastomosis between dorsal and ventral pancreatic duct, and this is the ventral pancreatic duct. So now what happens? Which are the parts that is now contributing in the formation of the major or main pancreatic duct? So look at that. It is the distal part of the dorsal pancreatic duct. This is the distal part of the dorsal pancreatic bud. Then this anastomotic channel between these two and the ventral pancreatic duct. This much region will form what? Your main pancreatic duct, and you can see it is opening along with the common bile duct into your major duodenal papilla. That is how you get your major pancreatic duct along with your common bile duct coming and opening into your major duodenal papilla. So it is the distal part of the dorsal pancreatic duct, then the anastomosis between these two, plus the ventral pancreatic bud along with the bile duct here, which is opening into the major duodenal papilla. And you know, just 1 or 2 centimeters above the major, you find the opening of minor duodenal papilla. In the minor duodenal papilla, you find the minor pancreatic duct is opening. So which part is forming the minor duct? It is the proximal part, this part, the proximal part of the dorsal pancreatic duct that is forming the coming and opening into the minor duodenal papilla. Fine. So that is how you get the duct system of pancreas. Now read that. 2 endodermal pancreatic buds at the junction of foregut and midgut. One is dorsal, the other one is ventral. And with the rotation of the duodenum, the ventral will come to the right. You know that. Dorsal will come to the left. Now what happens? This ventral will shift from the right to left and then come and fuse with the dorsal pancreatic bud. So now what happens? This upper part of the head, body and tail will be developing from the dorsal pancreatic bud, lower part of the head and the uncinate process from the ventral pancreatic bud. Fine. So I think with that you are familiar with the development of pancreas plus the duct system you have to mention, plus some applied aspect. One of the very common applied aspect you get here in pancreas is annular pancreas. You find this pancreas will be forming just like a ring, constricting the second part of the duodenum. And one of the very common sign, radiographic sign you get there is a double bubble sign, just like that of stenosis. Fine. So what is that embryological cause that causes the annular pancreas? That is a bifid ventral pancreatic bud. Usually you get a single pancreatic bud, and that will rotate to one side and then come to lie just below the dorsal. But in certain cases you find this ventral pancreatic bud will be bifid, will be two. So what happens, one will rotate in clockwise direction, the other one will rotate in an anti-clockwise direction and then come and fuse with the dorsal. So what happens, you get a ring-like constriction around the duodenum, and that is what is termed as annular pancreas. Fine. So that is first applied aspect. Second is divided pancreas. What do you mean by the term divided? Means there is failure of fusion of these two, ventral and dorsal pancreatic bud. That is what is termed as divided pancreas. Then you can also see the accessory pancreatic tissue present somewhere else, that is at the walls of the duodenum, Meckels, everything. So in certain regions you find accessory pancreatic tissue may be present in the gallbladder, the lower part of esophagus. There are certain regions where you find an accessory pancreatic tissue. Next is inversion of pancreatic duct. The main pancreatic duct will be formed by the duct of the dorsal pancreatic bud. Just the opposite. The main pancreatic bud or the main duct will be actually the dorsal pancreatic one, and the minor one will be the small one below. So an inversion, just the opposite of the normal, you get in an inversion of pancreatic duct. So this much applied aspect of pancreas. That is a picture showing annular pancreas. You find bifid pancreatic bud. This is the dorsal pancreatic bud. This is the ventral, that is bifid. And now look at it. It is rotating in two direction and then coming and fusing with the dorsal one and forming the annular pancreas. Fine. So with that we finished the derivatives of foregut. So I'm including spleen also here, not because it is an endodermal derivatives. Spleen is mesodermal, but for completion's sake, I'm including the development of spleen also here. But don't mistake it as endodermal or coming from the foregut. It is not like that. Spleen is actually mesodermal. Fine. So you find small masses appearing in the mesoderm as a mesodermal thickening present in the dorsal mesogastrium. Then what happens, all these small masses will finally join together to form the normal spleen, and that pattern of fusion is often represented as the presence of notches in the anterior or superior border of spleen. Fine. And what happens is, with the development of spleen in the dorsal mesogastrium, this dorsal mesogastrium will be split into two. I think I've already discussed it when I'm taking stomach for you. Do you remember that? With the development of spleen, that dorsal mesogastrium now gets split into gastrosplenic ligament and lienorenal ligament. Do you remember? Just look at that. What you see there is this is your spleen. It's a mesodermal thickening, or it's a derivative of mesoderm appearing in the dorsal mesogastrium. And with the development of spleen, this dorsal mesogastrium is now split into one ligament here, that is from the stomach to spleen. So what you call gastrosplenic. The other one from the spleen to the posterior abdominal wall, that is, it is going in front of the kidneys. So you call it as lienorenal ligament. Fine. So that is how you get the development of spleen. And applied anatomy, accessory spleen. Normal spleen, accessory spleen could be present in different locations. It could be present many sites such as hilum of spleen, gastrosplenic ligament, tail of pancreas, like that. So what is the clinical importance of this accessory spleen is that it may undergo hypertrophy after splenectomy. So even after splenectomy, the symptoms of the disease will be still persisting. That is because of this accessory spleen. Next is lobulated spleen. Normally I told you small masses appearing in the mesoderm. These mesodermal masses will join together to form your adult spleen. But in certain cases that lobulated pattern will be persisting. And that is what is termed as the lobulated spleen. So I think this much details you can remember. of spleen. So with that, we finish all the derivatives of foregut plus the development of spleen. So in the next session, we'll continue with the derivatives of midgut. So I think this much is clear to you. Fine. So I think I'll stop here and continue with the next session. Okay, thank you.